going to, witness in a couple of minutes. However, my group is working on different concepts like understanding the mechanism of Viper bites induced sustained tissue distraction at the site of bite. See here, before getting into these things, in case if you come across any doubt, you can intervene, definitely. You have to ask questions and I will be very happy to answer those in case if I know. So, understanding the mechanism of snake venom induced oxidative stress leading to hypoxia. And my students are also working on understanding the molecular mechanism of Viper bites induced nephrotoxicity and understanding venom neutralization strategies by anti-venom and small molecules, and understanding venom variability due to geographic distribution of snake species, and characterization of molecules of therapeutic importance from snake venoms. And these are all pertaining to snake venom related ones. And in case if you have any doubts regarding any of these things, you can ask questions. And in platelet biology, we try to understand the crosstalk between the mechanisms which favor platelet survival and the mechanisms which favor platelet death in various clinical conditions, particularly type 2 diabetes mellitus. So my current talk is focused on the first aspect, understanding the mechanism of Viper bites induced sustained tissue distraction at the site of bite. So based on this, so this is the title of the topic. Maybe I'll address for maybe another 50 to 60 minutes. Now, this is the global burden of snakebite. I think I can't see the title over here. 1 minute, please spare me. Madam, hello. Please. So something is blocking the titles of each slide. Just I want them to clarify this. It's
not visible clearly.
Hide. Oh, hide ಆಗ್ಬಿಟ್ಟು ಕಷ್ಟ. No. ಯಾರು ಇಲ್ವಾ ಈಗ?
ಇಲ್ಲಿದ್ರು ಸರ್ ಇಲ್ಲ ಟೀ ಹೊಂಟೋದ್ರು.
ಎಲ್ಲಿ ಇಡ್ಲಿ ಟೀ ಇರುತ್ತಲ್ಲ ಮೇಡಮ್, ಕರೀರಿ ಅವ್ರನ್ನ. ಎಲ್ಲಿ ಇದನ್ನ ರಿಮೂವ್ ಮಾಡ್ದೇನೆ ನನಗೆ ಆಗ್ತಿಲ್ಲ ಅದು. See, this are the titles. Based on this title, now it's fine.
ಇದು ಕರ್ಸರ್ ಅಲ್ಲಿ ಹೋದಾಗ ಸರ್ ಡೌನ್ ಆಗ್ಬಿಡುತ್ತೆ.
ಬಂದ್ಬಿಡ್ತಲ್ಲ, ಪುನಃ ಬಂದ್ಬಿಡ್ತಲ್ಲ.
ಅದೇ ಸರ್, ಈಗ ಕರ್ಸರ್ ಕೆಳಗಡೆ ಇರಬೇಕು ಸರ್. ಇಲ್ಲ ಈಗ ಹೋಗುತ್ತೆ ಸರ್. ಕರ್ಸರ್ ಆ ಪಾಯಿಂಟ್ ಹೋಗ್ಲೇಬಾರ್ದು, ಇಲ್ಲೆಲ್ಲಾರು ಇದ್ರೆ ಹೊಟ್ಟೋಗುತ್ತೆ ಸರ್. ನೋಡಿ,
ನಾನು ಇಲ್ಲಿ ಮೂವ್ ಮಾಡಿದ್ರೆ ಬೇಕೆ ಬಂದೇ ಬರುತ್ತಲ್ಲ ಅದು.
ಮೇಲಲ್ಲಿ ತೋರ್ಸಕ್ ಹೋಗ್ಲೇಬೇಕಾ ಸರ್?
ಇಲ್ಲ, ನಾನು ಈ ಪಾಯಿಂಟ್ ಹೇಳ್ಬೇಕಂದ್ರೆ ಹೋಗ್ಲೇಬೇಕು ನಾನು. This is the sorry for the inconvenience. This is the annual global burden of snake bite. According to World Health Organization's report, 2017 and 2019 report, about 5.4 million people are bitten by snakes, of which about 2.7 million people express clinical symptoms. That means they require clinical or medical interventions. Of these, about 0.14 million die and 0.4 million suffer from permanent disability or disfigurement. Strikingly, India is the biggest contributor for this number. According to a million death study by Suravira et al., about 58,000 people are believed to be killed in the Indian subcontinent alone. However, this number could be an underestimation of the reality, as most of the cases occur in rural, remote places, that is deep pockets, and never get into hospital records. Thus, India alone is contributing to the extent of about 30 to 35% of the global burden. That's the reason why India is popularly called snakebite hotspot. Venomous snakes possess a pair of venom glands, which are the modified salivary glands called Jacobson glands, and they are connected to a pair of modified teeth called fangs. During envenomation, that is during bite, the venom glands and the fangs work like syringe. They push the venom into the prey or human victims through a hollow space in the middle. Now the question is, why venom for snakes? Snakes use venom for two important purposes. Number one is for prey predation. That is, in the process of prey acquisition, snakes may use their venom to immobilize, to kill, and to digest the prey. Secondly, whenever snakes are in danger or in threat, they use their venom as a defensive armament to defend themselves against their enemies or against their predators. You can see over here, a tiny creature, spitting cobra, tries to defend itself against a huge structure, an elephant that I have cropped off by spitting its toxic venom. Now let me define snake venom. Snake venom is a complex, very, very complex mixture of predominantly enzymatic and non-enzymatic protein and peptide toxins. Interestingly and incredibly, most of the toxins are target specific. They do target vital organs, vital systems such as nervous, muscular, and circulatory systems. See, these three systems are tightly interconnected, interlinked, such that effect on one system invariably affect the other two, resulting in immobilization or killing. As snakebite is a true example of a simultaneous lethal assault on vital organs, vital systems of the prey animal or human accidental victims. Thus, snake bites is a true, very true medical emergency. So in case if you come across any doubt, you can ask me, you can interrupt and you can ask me, no problem at all. So for better clarity, better understanding, let me classify snake venom toxicity into two types. Number one is the systemic toxicity. Number two is the local toxicity. Systemic toxicity is the toxicity experienced by the entire body, where the vital organs, vital systems, as I said before, are being targeted. This is highly dangerous. In an untreated condition, this is fatal. People die because of this. In contrast, local toxicity is the toxicity experienced at the site of bite. It is characterized by the edema, that's swelling, hemorrhage, that's oozing of blood, blistering, that's bubbling, ecchymosis, change in color of the tissue at the site of bite, incredible pain, and massive tissue destruction like this. Now, what is the treatment for this? The treatment involves the intravenous administration of polyvalent antivenom, the only therapy available as of now to treat snakebite cases. If administered in time, the therapy can save the victims from the danger of fatal systemic toxicity. But unfortunately, the therapy fails to neutralize the local toxicity, that is, the toxicity experienced at the site of bite. So in this case, even after the person is saved from the danger of death, the local toxicity, tissue destruction at the site of bite would continue maybe a couple of days, couple of weeks, couple of months, couple of years, maybe lifelong as well. So in this case, the bottom line in this case is that we have somehow therapy available to manage the fatal systemic toxicity. But as of now, we have no therapy available to manage the local toxicity. These are the endemic snakes of the Indian subcontinent. Endemic means densely distributed or thickly distributed throughout the country. They are the cobra, reed, Russell's viper, and Saskia viper. So in the Indian subcontinent, the snake bite is generally attributed to bite by any of these four snake species. Therefore, these four are popularly referred to as Big Four. Of the four, these two vipers, Russell's viper and Saskia viper, are known to inflict a devastating, debilitating local toxicity. Now, just to give you the gist of the impact of local toxicity, here I'm presenting two case studies reported by Chipax and his group from an African country. So this is the Chipax publication. This is the first case report in which an 18-month-old kid was bitten by Echis vocillatus snake right in front of his parents while playing in front of his home. The kid was taken to the hospital and dosed with antivenom immediately. Fortunately, the kid was saved from the danger of fatal systemic toxicity. You can save the kid. But unfortunately, look at the local toxicity over here. This somewhere is the bitten side. The entire limb is swollen. We have blisters. We have massive tissue destruction. This swelling continued even till fourth day of antivenom treatment. So in order to release the pressure of the swollen limb, doctors had to operate this, and it became like this. And this was the next stage. And finally, after 11 surgeries and for 33 days, a group of doctors managed to treat this with skin grafting. This is how it was treated, and this was the final result. But after this, what happened? Was this a permanent remedy or was there a recurrence? We have no idea. This is the second case report in which a 13-year-old boy was bitten by a suspected Echis ocellata snake while trying to catch a rat in a burrow. So in this case also, the boy was taken to the hospital, given antivenom treatment. Fortunately, the boy was saved from the danger of death. But unfortunately, look at the local toxicity over here. This somewhere is the bitten side. The entire limb is swollen. You have blisters. You have massive tissue distraction. You have massive ecchymosis. And the doctors also, noticed the development of compartment syndrome. And here also, the swelling continued even after the boy was saved from the danger of death. So in order to release the pressure of the swollen limb, doctors had to make several incisions on third day, and it became like this. This was the next stage. And after 8 surgeries for 25 days, it has reached this stage. So in order to prevent further tissue decay, there are no other options but to go for amputation of this limb, the worst ever therapy one can think of. Thus, local toxicity appeared to be much more dangerous than the fatal systemic toxicity. It gives a sort of feeling in victims that instead of dying bit by bit, let's say die once. In this case also, it so happened that once the boy got to know that his limb is going to be amputated, it seems the boy disappeared from the hospital. Not only disappeared from the hospital, it seems he disappeared from his locality itself. Now, when he disappeared from the locality itself, you can think of his psychological status, psychological trauma that the patients, victims are undergoing. It's not only the victims. When the person disappears like this, you can think of the psychological trauma associated with the family itself. So family is going to be in deep trouble, definitely, thinking about the victim. And these victims, majority of the victims, actually, they are youngsters. They have the life expectancy could be 70 years or 80 years. So losing this kind of limb, losing limbs, and with this kind of a psychological trauma, the victims have to sustain, survive for so long. This is the difficulty associated with local toxicity. So in the Indian subcontinent, this snake, Echis carinatus, is also known to inflict a similar kind of local toxicity. Now the question is, why this persistent tissue decay? Why no treatment available despite scientific advancement? Despite scientific advancement, treatment is not available because the molecular mechanism of this macabre has not been understood. If you understand the molecular mechanism, probably it is easy to suggest the remedy for this. So in this line, different leading labs of the world have been working, pouring millions and millions of dollars, and even WHO also funded heavily on this project. People started working on this aspect maybe since 1930s and 40s. But at this stage, I'm extremely happy and delighted to tell you the fact that it is our group for the first time made a major breakthrough towards understanding the molecular mechanism and also suggested a simple remedy in a mouse tail model. However, the only clue people got to know all these years was the accumulation of lots and lots of inflammatory cells at the venom injected sites. So having known that information, we wanted to repeat that experiment in our laboratory. So we repeated that experiment. You can see over here, this is the mouse tail control tissue section. It's a skeletal muscle. You can see over here, clear-cut muscular striations characteristic of skeletal muscle. This is the control PBS treated one. And this is Echis carinatus venom treated tissue section. Here you could see over here, there are no clues for the presence of muscular striations. The entire tissue looks like the chopped one, the minced one. But if you observe it carefully, the tissue section, you can see over here, the entire tissue is studded or loaded with black or purple particles. So if you take this region, blow it up or zoom in, so now the black or purple particles are conspicuous, they are more visible. These are nothing but the polymorphonuclear leukocytes or the neutrophils. You know, neutrophils are the first line defense cells of the innate immunity. They are the most abundant phagocytic cells, and they are known to mount 3 levels of defense. They are the phagocytosis, that is engulfing and digesting the particulate material. If the neutrophils are unable to carry out phagocytosis, that means the external agent is larger, they are unable to phagocytose, then more number of neutrophils accumulate on the surface and they undergo degranulation, releasing lots and lots of granular substances. So those granular substances are basically they're cytotoxic in nature, antimicrobial or antibacterial in nature. So right on top of the foreign invader, these granular structures, the cytotoxic agents are being released, and thus the neutrophils are trying to kill the foreign invader through degranulations. So once the neutrophils got to know that phagocytosis and degranulation are not going to work, then they mount a different level of defense where they work like soldiers, where they're ready to die for the defense cause. They're ready to sacrifice their life for the defense by ejecting their decondensed DNA, decondensed chromatin through a process what we call netosis. So here, I think for the participants, I'm introducing an important concept of netosis. I think most of you are aware of phagocytosis and degranulation, but probably netosis is not featured in textbook now. And netosis is the another important defense event, the neutrophils exercise. So I'm introducing the concept of netosis. The whole idea of today's discussion is to introduce netosis to you people. Probably, so you can ask me questions in case if you have any doubts regarding netosis. So my further talk will be focused much on netosis. So netosis is an ancient bioweapon in innate immunity where neutrophils eject their decondensed DNA, the decondensed chromatin, the nets. Nets means neutrophil extracellular traps. So coated with lots and lots of antimicrobial, antibacterial or cytotoxic agents. You can see over here, neutrophil elastase, myeloperoxidase, cathepsin G. Likewise, people have identified over 40 different such antimicrobial or antibacterial agents. If you take the neutrophils are the popular granular cells. So they are loaded with the 4 types of granules. You are aware of the fact that, I think. So we have primary granules, secondary granules, tertiary granules, and secretory granules. And those granules, except secretory vesicles or secretory granules, rest of primary, secondary, and tertiary, they are loaded with lots and lots of antimicrobial or antibacterial agent. So this is the cartoon showing only one neutrophil is ejecting its decondensed chromatin or decondensed DNA in 2 dimensions. Just imagine in 3 dimensions, 100, 1,000, lakh, 10 lakh. So neutrophils are undergoing simultaneously this kind of, netosis. You can imagine now the impact of netosis. The nets released are going to be like a thick spider web. So thick spider web can entangle any particulate structure. Or it's like a miniature cotton plug. It can block any blood vessel or prevent the blood flow. That is the impact of netosis or net formation. This novel mechanism of net formation or netosis was discovered by Walker Brinkman and Arthur Jasinski from Max Planck Institute, Germany during 2004. They published this novel finding in a top-notch journal, Science, during 2004. See, actually, the whole story of netosis was started with this initial finding. So 4-ball myristate acetate induced neutrophil autotoxicity. This was observed way back in 1980 by M.F. San from the John Hopkins Medical Institution, Baltimore, USA. So this was the first report regarding this in this line. And later, maybe after 11 years of gap, one more study came in from a group from Portugal. Activation of human neutrophils by 4-ball ester decreases the cytoplasm compactness and the lactoferrin content of the granulocytes. So this is another story came in. in 1991. And later, after 5 years, one more study from Japan, like rapid killing of human neutrophils by the potent activator 4-ball 12 myristate, that is PMA, accompanied by changes different from typical apoptosis or necrosis. So here they mentioned clearly apoptosis and necrosis. So this kind of PMA induced autotoxicity of neutrophils was different from apoptosis and necrosis. This was the study reported by Japan, a group from Japan. So these were the important studies. So 1980, 1991, and 1996. Then till 2004, it was remained, almost dormant, that particular area. Only during 2004, the actual mechanism of how the toxicity is caused to the neutrophils by PMA was discovered by Walker Brinkman and Arthur Zisinski from Max Planck Institute, Germany. So in our study, as I already mentioned, we could notice that at the site of Echis carinatus venom injection, you could see the accumulation of lots and lots of inflammatory cell, that is neutrophils. So venom can recruit lot many neutrophils at the site of injection. So you can see over here. So this made us to ask an important question. Are neutrophils connected to Echis carinatus venom-induced tissue necrosis? So you see, so initially I told you Echis carinatus venom is known to inflict, massive tissue decay at the site of bite. This is what, since Echis carinatus venom recruit neutrophil at the site of injection or at the site of bite. So we ask the question, are neutrophils connected to Echis carinatus venom-induced tissue necrosis? If they are connected, how are they connected? So we wanted to address this important question. So in order to address this, what we did was we isolated human neutrophils. So these are the control human neutrophils stained with hue stain. And this is the observation done in a fluorescence microscope. So these are the control neutrophils. You can see over here clearly the neutrophils in the resting state. They have, they clearly see the multi-lobed nucleus. So compared to rest of the cells, the nucleus is actually rounding or spherical in shape. But in this case, the nucleus is actually, it has no proper shape. The nucleus is actually divided into different lobes. That's the reason why. So neutrophils are also called polymorphonuclear leukocytes. Morphologically, nucleus is actually exists in different forms. So we have multi-lobe nucleus. So that's the characteristic feature of the neutrophils. So these are PBS treated neutrophils, and this is dose-dependent venom treatment. After venom treatment, let's say Echis carinatus venom 5 microgram per ml, incubated for 180 minutes. You can see over here, some of the neutrophils have already ejected their DNA fibers, and many have lost their, nuclear shape. They are, taken up a round shape here. So you can see this is the same thing, black and white, and it's colored. So this is with 10 microgram per ml of Echis carinatus venom. You can see over here, almost all neutrophils, they are programmed for netosis. You can see a change in shape of the nucleus. Many have ejected their decondensed DNA. See here, you can clearly see the nets released are like appear like spider web. So a thick spider web. And with 50 microgram per ml, you can see over here, almost all neutrophils ejected their chromatin, that is decondensed DNA. It is almost like thick spider web or a thick cotton plug. It can plug any blood vessel and prevent the blood flow. So this is what we just imagined, presumed. So this is a positive control. For any experiment to do, you need to have a positive control with you. So to get the validation of your experiment, the positive and negative controls are must. This is a positive control. Negative control, anyway, we have to keep one more thing that we have not kept here, but PBS we have used as simply a control one. So this is a PMA, 4-ball myristyl acetate. Remember, this discovery of netosis started with the PMA effect on neutrophils. So we've used PMA, 4-ball myristoyl acetate was used as positive control over here. So you can see over here, 4-ball myristate at just 50 nanomolar concentration, very minute concentration, it could induce net formation over. This is the black and white picture, and this is the colored one, the same thing. This is how we quantitated nets. What we did was we took neutrophils, added venom or PMA, incubated for 180 minutes, centrifuged, took the supernatant. Supernatant, this is how the supernatant, the DNA, the nets present. So nets are coated with lots and lots of antimicrobial, antibacterial agents. You remember that? So let's say here we have used neutrophil elastase myeloperoxidase DNA complex to quantitate the released nets. So this is actually dose-dependent effect, 5, 10, 15, 20, 25, 50 microgram. With increasing dose of venom, there is an increase in the nets. And this is the time-dependent effect. Till 180 minutes, we followed the time-dependent release of the nets. And this is the principle employed. So we have here, we have followed a DNA, MPO DNA capture LSA. MPO means, NE means neutrophil elastase, MPO means myeloperoxidase. So, I told you, we have the neutrophils are loaded with granules, primary granules, secondary granules, tertiary granules, and secretory vesicles. The primary granules are loaded with neutrophil elastase and myeloperoxidase. These are important, agents play a role in defense event, that is in innate immunity. If you understand innate immunity, the role of neutrophil elastase, myeloperoxidase, you will get, I think you get a better clarity. So what are the effector agents for nets formation? Hello, am I okay? Am I audible?
Yes, sir, you're audible.
Yes,
You're
sir.
audible, sir.
Yes, sir. Yes, sir.
So you have no questions to ask in between. Anyway, we can ask question probably later. So to induce nets formation, to have nets formation, what are the effector agents? How does net formation take place? So in this case, actually for net formation, you have the neutrophil elastase is very critical. That's present in primary granules, and also PAD4 enzyme. PAD4 is otherwise called peptidyl arginine deaminase 4. This is an isoenzyme 4. Peptidyl arginine deaminase, there are 5 different isoenzymes are there. The 4th isoenzyme is actually present in the neutrophils, that is in the cytosol of the neutrophil. And this is an important enzyme, and this is a calcium dependent enzyme. If one molecule of peptidyl arginine deaminase can bind 5 calcium ions at a time. So thereby, in presence of high calcium ion cytosolic calcium ion concentration, peptidyl arginine deaminase is getting activated. Okay, so neutrophil elastase and peptidyl arginine deaminase are very critical for netosis. See this one, neutrophil elastase along with myeloperoxidase. See this complex, neutrophil elastase and myeloperoxidase, this complex alone or in combination with PAD4 or PAD4 alone. So together or independently, these would induce nets formation. See here, this complex, neutrophil elastase and myeloperoxidase. I don't know how, we don't know. So far we have no answer. Like myeloperoxidase is always associated with neutrophil elastase. Okay, if you knock off myeloperoxidase activity, it seems neutrophil elastase activity is also lost. If you knocked off neutrophil elastase activity, even myeloperoxidase activity is lost. So how these two related, we don't know. But this complex is essential, and PAD4 is also essential. So this complex, neutrophil elastase myeloperoxidase complex and PAD4 independently or together, can induce nets formation. Okay, so we know that the entire story of netosis started with the PMA-induced autotoxicity of neutrophils. So just to have a better clarity on this aspect, I just summarized how PKC, 4-ball myristoyl acetate induce neutrophil autotoxicity or induce net formation. See, PMA 4-ball myristyl acetate, you know that it's a plant component, it's a non-hydrolyzable one, and it's an activator of PKC, protein kinase C. So, you know, protein kinase C in vivo, so it's a serine threonine kinase, protein kinase C. And in vivo, this enzyme is activated by, activated as a result of PI turnover. The PI turnover due to the activity of phospholipase C enzyme, we have IP3 and diacylglycerol are getting released. IP3 and diacylglycerol. So IP3 is actually hydrophilic in nature, and it has a different function. It helps in calcium mobilization from the internal stores. But PKC is activated by the diacylglycerol that is still associated with the plasma membrane. So the PKC in a calcium ion dependent manner translocates to the plasma membrane, where it binds to diacylglycerol and get activated, the PKC. So this PKC, that active PKC, use variety of cellular proteins as its substrate. So it phosphorylate on specific serine and threonine residues of various cellular proteins. And one of the important substrate of PKC is the NOX enzyme. NOX, otherwise FOX, PHOX. In NOX means NADPH oxidase, FOX means phagocytic oxidase. So both are same. So PKC will activate NOX enzyme. NOX enzyme is responsible for generating ROS. So NOX enzyme in an NADPH dependent manner, it partially reduces oxygen to O2 dot. So superoxide radical is formed. That is the reactive oxygen species, most important reactive oxygen species. The ROS that is generated, superoxide radical that is generated, immediately it is converted into what? Anybody answer? Immediately it's converted into superoxide radical O2 dot. If it is reduced by single electron, it is superoxide radical. So it's immediately converted into what? It is converted into H2O2 by superoxide dismutase enzyme. So H2O2 is also an important ROS, reactive oxygen species. Unlike other reactive oxygen species, H2O2 can easily diffuse through membranes. It can reach to various subcellular compartments, and also it can go out of the cell depending on the concentration. So if the cell intracellular H2O2 concentration is more, then it can easily diffuse out of the cell through the plasma membrane. It is the only ROS can easily cross through plasma membrane or membrane. So H2O2 is formed. So in an H2O2 dependent manner, I told you we have the primary granule in neutrophil. It is also called azurophilic granule. So in primary granule is noded with neutrophil elastase and myeloperoxidase and various other cytotoxic agents. And in an H2O2 or ROS dependent manner, the neutrophil elastase and myeloperoxidase translocate out of this azurophilic granule and gets into the cytosol. So neutrophil elastase, it's a protease, it's a serine protease. Serine is there in the active site, like chymotrypsin. So it's a serine protease. So it gets into the cytosol, and in the cytosol, the most important thing is that it would degrade variety of cytosolic proteins, and some studies say that it would act on F-actin. So F-actin, that is fibrous actin. Fibrous actin dynamics is very critical for a cell for its mobilization. See, all your phagocytes, they move from one place to a different place through stretching their pseudopodia. So stretching this of pseudopodia is mainly controlled by the actin dynamics. We have, you remember actin, we have F-actin and G-actin. We have loading and unloading continuously take place. So when it wants to stretch the pseudopodia, this dynamic event has to take place. loading and unloading, and as a result, it will just pseudopodia is being stretched. So cell will move from one place to a different place, maybe through a chemotactic mechanism. So here, through this mechanism, cell will move, and in this case, what happens is neutrophil elastase. Once it gets released into the cytosol, it seems it act on the actin dynamics. It would cleave F actin, and when F actin is cleaved, we have no more actin dynamics exist. As a result, cell will get arrested at the place where netosis is required, because it can't form pseudopods. It cannot stretch pseudopodia and move from one place to other place. So some studies would say neutrophil elastase would degrade F actin, and as a result, it would arrest cells at the place of where netosis is required. Okay, and some studies say the effect in dynamics is altered in the absence of even neutrophil elastase. It won't require
Sir,
neutrophil elastase. Yes.
can you please explain once more this entire process which you are explaining, the mechanism?
Oh, that's wonderful. Anyway, you want me to explain? Okay.
Once again,
One
because
this PMA induced
yes, sir. Because few things are not clear to me. So can you please explain once more?
Yeah, I will do that, no problem. So this is what the rest of the things, okay, those are all research oriented. I just wanted to introduce the net formation here. That's fine. Okay, if you understand this much, that's good enough. So PMA causes autotoxicity, right? It kill neutrophils. So people wanted to understand the molecular mechanism. It's actually author Zisinski and Brinkman. They worked extensively to understand the molecular mechanism. So PMA 4-ball myristyl acetate, it's a non-degradable activator of protein kinase C. PMA can easily get into the cell. It can cross the plasma membrane. It can get into the cell. Remember, understand? PMA can easily get into the cell. That is, PMA means 4-ball myristyl acetate. 4-ball myristyl acetate can easily cross the plasma membrane, get into the cell. And it is the activator of protein kinase C. Protein kinase C, okay?
Yes, sir.
So, protein kinase C is an important kinase. It's a serine threonine kinase, as you are aware of. And it is activated, especially during signal transduction. Wherever we have PI turnover take place through phospholipase C-mediated PI degradation, we have the products diacylglycerol and IP3 are released. Remember, so IP3 is hydrophilic in nature. It translocates, it comes down and act on endoplasmic reticulum. It acts on calcium ion channel. So that's the gated channel. It binds to the calcium ion channel in the endoplasmic reticulum, smooth endoplasmic reticulum. As a result of IP3 binding to the calcium ion channel, the channel opens up. As a result, the calcium ion that are stored in the endoplasmic reticulum now comes out to the cytosol. As a result, there is an elevation or increase in intracellular calcium ion concentration. That's one thing. And in a calcium ion dependent manner, PKC has EF and loop, right? EF and loop. So EF and loop is important for calcium binding. PKC has that EF and loop, so it binds calcium. And once it binds calcium, PKC get activated and it translocates into the plasma membrane, where in the plasma membrane diacylglycerol is going to be present. Remember, diacylglycerol is present. Remember, through phospholipase activity. So now PKC goes and binds to diacylglycerol. Now PKC gets activated fully. So for PKC activation, normal PKC activation, we require diacylglycerol and calcium ion. Okay, for normal PKC activation. But PMA, in presence of PMA, it does not require anything. It does not require calcium ion. It does not require diacylglycerol. PMA can directly activate PKC enzyme. So PKC, once it is activated, it can phosphorylate wide variety of intracellular proteins on serine and threonine residues. Have I made it clear? Okay.
Yes, sir. Yes, sir. Clear, sir. You go ahead, sir.
Phosphorylates, and one of the important substrates is actually NOx enzyme. NOx means NADPH oxidase enzyme. It's a very complex enzyme. Some of the subunits are associated in the plasma membrane and some present in the cytosol. Okay, so when in this condition, NOx enzyme is actually getting activated, where PKC phosphorylates one of the subunits of NOx, and after phosphorylation, we have NOx enzyme is getting activated. Got it? PKC activate NADPH oxidase enzyme. So NADPH, the active NOx enzyme, its function is to reduce oxygen, generate ROS. So it partially reduces oxygen by 1 electron, and you get superoxide radical. Reduction of oxygen by 1 electron, you get superoxide radical. And superoxide radical is highly unstable. The moment superoxide radical is formed, we have superoxide dismutase enzyme. Immediately that convert that superoxide radical into hydrogen peroxide. Okay. So now in a hydrogen peroxide, that's also an ROS. In an ROS dependent manner, now in an ROS dependent manner, neutrophil elastase and myeloperoxidase, they were residing in azurophilic granules initially, now translocate out of the vesicle through an unknown mechanism. How they translocate out of the granule, nobody knows, because even after translocation, the granule was found to be intact. There is no lysis of granule. Granule was intact, but neutrophil elastase and myeloperoxidase, they just translocate out in an ROS dependent manner. Because if you block ROS formation, neutrophil elastase and myeloperoxidase will not come out. It in an ROS dependent manner, it comes out, and neutrophil elastase is a serine protease, remember, and it's a very active protease. It would use variety of intracellular proteins as its substrate. It would cleave variety of substrates. Among them, effectin could be one among them. Effectin. So it would degrade effectin. As a result, effectin dynamics is critical, as I said, for the mobility of these neutrophils from one place to the other place through stretching pseudopodia. So effectin loading and unloading is very critical. So here, since neutrophil elastase is degrading effectin, the cells won't move from that place. So they are just be placed or arrested at place where netosis is required. Okay, now is that clear? Clear?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
After having done this job, neutrophil elastase and myeloperoxidase, this complex, in an unknown mechanism, they translocate into the nucleus. Remember here, they translocate into the nucleus. Their molecular mechanism has not been understood. So gets into the nucleus. In the nucleus, we have chromatin. Remember, we have H2A, H2B, H4, H3. So nucleosome is there, right? Is it not?
Yes,
So
sir.
in this
Yes.
nucleosome, we have histones, right? H2, H2B, H4, H3. These are histones, and they help in actually compactly packing the DNA. Is it not? Now, histones are proteins. Neutrophil elastase is a protease. The neutrophil elastase, so in the nucleus, preferentially degrade H4. It will degrade preferentially H4, and then it would degrade other histones also. As a result, what happens is this nucleosome structure is getting destabilized. You understand this? Now, since histones are degraded, now what happens is the positive charge on DNA increases. As a result, they opens up. So negative charge on DNA, not positive charge on DNA. Negative charge on DNA, opens up. Negative charge, negative repulsion take place. As a result, unwinding take place. So now the DNA unwinding take place. So we have DNA fibers are formed, or chromatin is formed. So that increases the size or the volume or pressure of the nucleus. You understand this? As a result, this increase in pressure will cause the opening of this nucleus, or through some mechanism that increased chromatin in the form of vesicles, they may be ejected out of the cell. Ejected out of the cell, this chromatin threads, the DNA threads are ejected out. This is one where neutrophil elastase is participating. On the other hand, 4-ball myristoyl acetate, PMA, was also known to induce stress on endoplasmic reticulum. So when it induces stress on endoplasmic reticulum, you know, endoplasmic reticulum stores a lot of calcium ions. As a result of stress, calcium ion is released from the endoplasmic reticulum. So the increased calcium ions now activate PAD4 enzyme, peptidyl arginine deaminase enzyme. I told you peptidyl arginine deaminase, it can bind 5 calcium ions at a time. So once it binds calcium ions, so peptidyl arginine deaminase PAD4 enzyme getting activated and it translocates into the nucleus from the cytosol. So now PAD4 enzyme gets into the nucleus and where we have again PAD4 enzyme act on again histones. So it would preferentially use H3, histone 3, then on other histones also. What it does is initially on H3, that histone 3, is that realizes the hydrolytic deamination of peptidyl arginine. We have peptidyl arginine here in histone. We have arginine. Remember, arginine has a side chain. This side chain is actually what we call guanidinium group. It carries positive charge at neutral pH. This is important. So peptidyl arginine, so is used by this PAD4 enzyme. It carry out hydrolytic deamination of this nitrogen present in NH2 group present in the guanidinium group, and this becomes peptidyl citrulline. So look at this. In this case, the positive charge on the histone 3 is now lost. There is no positive charge here. It becomes H3CIT, citrullinated histone 3. Histone 3 becomes citrullinated histone 3. We have citrullination here. This is a citrulline. No guanidinium group means this is citrulline. Citrulline is actually not a real amino acid of it's coded by any codon. It's not coded by any codon. But now because of this chain, citrulline is there. Okay. This enzyme will act only on peptidyl arginine, not on free arginine. That you have to remember. It uses peptidyl arginine, H3 only, and after H3, it uses other histones also. As a result of loss of a positive charge, now negative charge on DNA, density increases, negative repulsion take place. As a result, DNA unwinding take place. As a result, again, nets are extruded out. This is how the molecular mechanism where the PMA induced, nets formation you can think of. Am I made it clear? Hello?
Yes, sir.
Yes, sir. Clear, sir.
Okay. Okay. So this is what the mechanism is. I think my job is done. I wanted to just inculcate this netosis mechanism. See, the netosis mechanism, as I've already said, it would, neutrophil elastase itself does, or PAD4 itself does. Together, they may promote netosis. Independently or together, they promote netosis. So, for netosis, these two enzyme systems are critical. I told you, like, see, here, while talking about the F-actin, disorganization of F-actin. So here, you see, there are studies indicate that F-actin is getting degraded, but there are studies contradictorily. They say F-actin is not degraded. Instead, F-actin undergo disaggregation. So F-actin becomes, G-actin. The polymerization is going to be inhibited. This is what we have, evidence for both degradation as well as depolymerization. So here just I am showing how F-actin disappears during net formation. So in this case, actually this is not Echis carinatus when I'm induced depolymerization. I'm just presenting depolymerization of F-actin induced by a cardiotoxin fraction from Indian cobra venom. So from Indian cobra venom, we have isolated a cardiotoxin fraction, and that also induced strong netosis. That is not a protease enzyme. Okay. So see, this cardiotoxin induced strong netosis, and you can see over here how the F-actin dynamics is getting altered. So you can see over here, these are the intact cells, neutrophils. You can see clearly the blue one, nucleus has been stained. Red one is actually the F actin has been stained. This is the principle employed. We have used phalloidin coupled to Alexaflor 488. So this Alexaflor, when you excite, at 488 nanometer, they emit at 496 nanometer the red color. So you can easily monitor like this. See, these are the control cells. See, look at here with cardiotoxin fraction 10 microgram per ml, just 10 minutes. You can see the cells have been programmed for nets formation. The nucleus shape has gone, and you can see clearly the gradual disappearance of the red color. Intensity of the red color is coming down. So you can see our nets, predominant nets here, and the disappearance of red color here. So that means F actin is getting disappeared. That color is getting disappeared. That's suggesting that either it is subjected for proteolysis or it undergo depolymerization. So 10 microgram, 30 minutes. Look at this at 30 minutes, you can see this. 10 microgram, 60 minutes, almost all red color are gone. Yeah, the red color is gone. And it is just started with the nets here. So this nets formation induced by this cardiotoxin fraction was not sensitive to protease inhibitors that are neutrophil elastase inhibitor or serine or metalloprotease inhibitors. So even in the presence of protease inhibitors, this happens. That means this disappearance of F actin is not proteolytically triggered. It is not cleaved or disappeared. It is through disaggregation. It happens. Okay. And this is another control cells we have, but if you use actin polymerizing or depolymerizing inhibitors, we have inhibitors, they can block actin polymerization, and we have inhibitors, they, block depolymerization. So actin dynamics can be inhibited by, we have specific inhibitors are available, but in presence of actin polymerizing and depolymerizing inhibitors, there was no netosis of CTX induced netosis, no netosis here. So suggesting that, during netosis, we have F actin dynamics is drastically, altered. You understand this? Hello? Understood?
Yes, sir.
Yes, sir.
And rest all we wanted to just we wanted to study the Echis carinatus venom induced netosis here. See, you can see over here unstimulated cells. You can see they're smaller in size. Unstimulated cells, no signal for H3C80. This is overlay. See, you can see over here Echis carinatus venom stimulated cells. Their size is increased. You can see over here there is a lot of signal for H3C80. This is the overlay. This is the positive control PMA. 4-palmyristyl acetate. Again, cells are programmed for netosis. You can see a lot of H3C80 overlay. And this is the principle employed here. Okay. And this is, as I said, H3C80 is formed. It's Echis carinatus promote H3C80 formation. That is important for, that is arise due to PAD4 activity. So we have to demonstrate the PAD4 expression here. So you can see over here, PAD4, control unstimulated cell, feeble signal, PMA induced, we see a lot of signal. With increasing concentration of venom, we have a lot of PAD4 signal. This is GAPDH, is actually loading control. So PAD4 will generate H3C80, citrullinated gestone 3. You can see unstimulated, PMA, we can signal. With increasing concentration, we have a lot of H3C80. H3 was used as loading control here. This is the density, corresponding densitogram here. So based on this, we came up with a hypothesis that Echis carinatus venom induced netosis, nets block blood vessels, and also sequester venom components at the site of injection. As a result, we have compartment syndrome is developed, and this blocking blood vessels and sequestering venom components induce a lot of physiological changes at the site of bite, like starvation, hypoxia, accumulation of metabolic weights, extracellular matrix degrading enzymes, MMP is getting activated, venom enzymes are involved. We have cytotoxin. They're all like, they do the job of tissue destruction. This is what the hypothesis we proposed. And we wanted to show the same in a mouse model. So that's in vivo studies. This is PBS injected mouse tail, you can see clear mouse tail. This is Achis canadas venom injected mouse tail. After 8 hours, it appears like this. See, at 8 hours, probably this part of the tissue is already dead and gone. You have used LD50 of Achis canadas venom 1 milligram per kg body weight. So this is again control mouse tail. First day, it's all like this. Second day, fourth day, sixth day, 10th day. See, simply we've allowed to stay, since we are doing for the first time, we've allowed to stay the, the tail like this. We followed for 10 days. Say at 10 day, it detached. See, at first day itself, it is dead and gone. But we simply allowed it. See, this is a control one, Achis canadas venom injected one. So in 8 hours of tissue, we have followed the H3C80 and MPO activity. Don't have to worry too much. So we want to demonstrate the blocking of blood vessels. We proposed the hypothesis blocking of blood vessels, sequestration of venom components at the site of injection, right? So we wanted to demonstrate the blocking of blood. See, this is the mouse tail tissue section. This is the blood vessel actually. The artery, you can see the accumulation of lots of neutrophils. Neutrophils were stained with specific marker LY60. LY60 is the specific marker, and we have used marker for locating this, and we have corresponding H3C80. This is DNA. This is overlay. So we have the principle. So thus. Neutrophils recruit, Achis canadas recruit neutrophils, and as a result, recruit neutrophils, trigger neutrophils for netosis, nets accumulate in the blood vessel, block the blood vessel, and prevent the blood flow. So this is again lactoferrin, another marker for netosis, H3C80. So you can see over here in tissues. So I also said venom. Nets would sequester venom components. See, neutrophils, DNA, we have treated with the antibody for Achis canadas venom. We have a lot of signal for, the DNA and interacting Achis canadas venom. You can see this is the overlay. So thus, DNA binds the venom component strongly. So because most of the most toxic toxins of the venom are positively charged at neutral pH, and the DNA is negatively charged at neutral pH. We have charge interaction take place. So wherever DNA is there you see the binding of venom components. This is the overlay, and this is also DNA and venom interaction was demonstrated through gel retardation assay. It's a mobility shift assay. See these the markers of DNA. And this is the PCR product of DNA. And see, PCR product alone moved to this extent. 160 base pair DNA fragment we have used, moved to this extent. In presence of venom, see, look at this, the mobility has been retarded. So indicating that we have the DNA and venom components interact strongly. So thus, venom DNA, that means chromatin, or DNA fragments sequester venom at the injected site. This is the principle. And see, why we have stable nets induced by Achis canadas venom is because Achis canadas venom is devoid of, is lacking in DNase activity. So this is the control. This is the positive control, the pancreatic DNase. And this is Achis canadas venom 50 microgram, 100 microgram, 200 microgram, 500 microgram. No sign of DNA degradation here. So Achis canadas venom is lacking DNase activity. So as a result, what happens is this is the control mouse tail tissue. Achis canadas venom alone, so you can see the accumulation of venom in the tissue. But supposing if you incubate, so if you inject Achis canadas venom along with DNase enzyme, you don't see the accumulation here. So this is the principle employed here. So thus our hypothesis was demonstrated, blocking of blood vessels, sequestration of venom toxins, and all these things in tissue destruction was demonstrated. And we asked a question, can we treat this? What if we cut the DNA fibers with chromatin? This is what hypothesis, Achis canadas venom, induced netosis, nets block blood vessels, sequester venom component leads to tissue decay, degradation. Supposing if you use DNase and DNase would degrade the nets, that is chromatin threads or DNA threads. As a result, no block of blood vessels. As a result, no tissue decay. Wanted to demonstrate that. So this is again the same experiment. This side already showed this. Control 8 hour. See, 3 hours after venom injection, we gave a DNA shot here. 3 hours after venom injection, we gave a DNA shot here. So you can see over here, this tissue, the tail tissue, is gradually, this first day it appeared like this. Second day, it almost became normal. Fourth day, it became normal. Tail has been rescued here. So this is what the same experiment. So control, this is Vekis carinatus venom at the site, it accumulates and degrades the tissue. Initiate netosis, accumulate there, degrade the tissue. But if you treat with DNAs, no accumulation here, all gone. Okay, these are some data. The snake venom DNAs, for the first time we have demonstrated its important role in lethal toxicity. So before our study, people have been working on DNase enzyme, snake venom DNase enzyme since several decades, maybe 1930s, 20s, like that. So once they started using antivenom or started characterizing snake venoms. But till our study, people thought DNase enzyme has nothing to do in the toxicity of the snake venom. People thought it just play a role in digestion of the prey or digestion of the food. Whatever DNA components are there in the prey that is in the food, DNA is used for digestion. So it's our study for the first time demonstrated the significant role played by the DNase in venom lethality. Its presence can make the venom more toxic or less toxic, or more lethal or less lethal. So I've been talking about DNA, the nets. Why nets is so important? Because, it's nets like, they're compactly packed in a small, tiny space. See, look at this. We have about, our haploid genome, about more than 3 billion base pairs. Neutrophils being deployed, we have 6 billion base pairs. So those 6 billion base pairs are involved in creating 46 pieces of chromosomes. And those 46 pieces of chromosomes, combined, they aggregate and they compactly packed with the help of histone and compactly packed in a small place, nucleus. Is it not? Your neutrophil itself is a small, tiny structure. Within the neutrophil, we have even more tiny structure, nucleus. Within that nucleus, this 46 chromosomes are packed. And if you take all 46 chromosomes, unwind the DNA, place one after the other, and measure the length of it, it would come to around 2 meters long. That means 2 meter long DNA and even above is compactly packed and placed in a tiny space. Now imagine, it's like this. Say we have thread here, cotton thread here. Maybe if I unwind this, probably it would measure about maybe 300 meters. It's so long thread, but compactly packed in this tiny structure. So supposing if we unwind this, you can see the bulkiness of that. It spreads everywhere, and this space is not enough to occupy that one. So it's exactly the same way. 2 meter long DNAs. Now, let's say each neutrophil has the ability to eject 2 meter long DNA. Imagine 10 neutrophils, 100 neutrophils, 1,000 neutrophils, 10,000 neutrophils, 1 lakh neutrophils, 10 lakh neutrophils. If every neutrophil is ejecting 2 meter long DNA, now you can see the impact of the nets that's being ejected. It is definitely like thick spider web that can, encapsulate all particulate structure, won't allow particulate structures to penetrate through, or it is like a thick cotton plug, miniature cotton plug, and it can plug any blood vessel and prevent the blood flow. So this is the picture. See, look at that. This is the picture shown by the authors, Brinkman author Zisinski, where Shigella flexneri parasite parasitic bacteria, disease-causing bacteria for the intestine causes diarrhea and all. This red color bacteria, rest all, nets. So entrapped in the net, and this is easily cleared by phagocytic system. So thus, in summary, Achilles carinatus venom induces robust net formation. Nets block blood vessels and prevent blood flow, leading to tissue distraction. NET's degradation by DNase1 reestablishes blood flow, hence suggests a mode of therapy. Our lab was first to establish NETs and DNase role in venom toxicity, established the trial model to study tissue necrosis. This study was the first to uncover the possible mechanism of Echinus venom-induced tissue destruction. This study may potentially save lakhs of limbs annually. And so this is the publication we got. So actually, this should have been gone to even bigger journal, but there are so many hindrances are there. But anyway, this Nature Communication itself is a huge journal for a person working in universities, struggling for even distilled water. So this is most important. A few days before publication, the NPG, Nature Publishing Group, press office distributes a press release about upcoming Nature Communications paper. I am pleased to inform you that your paper has been selected for highlighting. So we did not go for press release. Nature Publishing Group, that press itself, they themselves went for the press release. That's World Press, and it got featured in so many daily news journals. And most important one is it also got featured in National Geographic. So featuring in National Geographic is probably much bigger than in a publishing paper in Nature Communication, probably. So this is the impact of our study. This is my wonderful group. And this is the PhD work of this guy, Gajanan Katkar. Along with me is the Professor K.S. Girish. He was my PhD student and still associates with me. And these are all, contributed for this study. And these are my mentors. T.V. Gowda, with whom I did my PhD. Before that, I worked with Professor P.S. Sastry in Indian Science. And he introduced me for the research. And Professor Ram Rajshekharan was my senior in